Good morning. My name is Stephanie Elena, VP of Marketing at KromaTiD. I would like to welcome you to our webinar, Combining Synthetic and Evolutionary Biology Reveals DNA Therapy to Combat Cancer Chemoresistance. In today's webinar, Dr. Gábor Balázsi, a Lawfer professor at Stony Brook University, will be discussing his latest publication and provide an overview of the KromaTiD assay that was used as proof of concept in this study. Accompanying him will be Aaron Cross, our VP of Research and Product Development, who will give an overview of DGH assays and the role our data plays in oncology research. At the end of the webinar, we will have our Q&A session. However, if you have any questions during the webinar session, you can put them in the Q&A box to be answered at the end. Starting our introduction today will be Christopher Tompkins, our CTO here at KromaTiD. Now I will give the floor to Chris.
Thanks, Stephanie. I'm Christopher Tompkins, CTO here at KromaTiD, and today I'd like to welcome all of you to the first of our 2024 webinar series. Today we're honored to have Professor Balázsi join us as a guest speaker to discuss his work in synthetic biology. Professor Balázsi has developed methods for tuning gene circuits that expand beyond simple knock-ins or knockouts, in essence installing dimmer switches into the genome to provide not just the correct proteins, but also the correct amount of the correct proteins. Following Professor Balázsi today, our own Aaron Cross will join us to discuss DGH applications and how we supported Professor Balázsi's R&D efforts. And now, Professor Balázsi.
First of all, I'd like to thank everyone who made time to hear this webinar at this hour, wherever you are, welcome. And what I'd like to talk to you about is combining two areas of biology that you may or may not have heard about, or even if you heard about, you've never heard them combined. So on the right side of the slide, you see this colorful picture. This is the picture of a chromosome pair. This is a package in which DNA, our hereditary material, is packaged. So the DNA that carries information on how our cells work, how we are, is packaged in these chromosomes inside every one of our cells. And I'm showing this because it's going to be a recurrent theme in this presentation. All right, so to motivate today's talk, I'd like to talk about controlling cells. And I'd like to propose that controlling living cells is a 21st century challenge. And you might have seen this movie, some of you, titled Gravity. It is a story about space travel and being alone in space and figuring out how to get back to Earth. The essence is that a person sitting in a spaceship, manipulating buttons and control knobs, is able to control spaceships, reposition them, stop them, restart them, and so on. And all of that is due to the ability of controlling non-living systems. And we all know that the major accomplishments of the 20th century were to learn how to control non-living systems. And we see obvious consequences. There was a man on the moon. We have actually spaceships, satellites that we can control. And we actually have a Mars rover walking on the face of another planet and submitting images, taking samples, and so on. So all of this is due to advances in the 20th century where we learned how to do proper electronics, mechanics to control non-living objects, systems. However, I would like to say that even though we are able to do all of this, we are still unable to control living cells. And that would be the challenge of the 21st century, this century. So imagine we now can have control knobs and switches, which we manipulate and then control living cells, telling them to divide, to move, to stop moving, to stop dividing, to differentiate, and so on. Why would that be good? Well, it would be good for two reasons. First of all, it would be good for better medicine. Because if you think about it, most of today's medicine relies on killing cells. So we want to kill cancer cells. We want to kill infectious cells, bacteria. That's a very rough and primitive way of control. I think we can do better, and there are signs of this. Gene therapy is advancing, and I'd like to say that synthetic biology has possibly a major role in this. So controlling cells would give us ability to do better medicine by creating cells that produce insulin, that stop being cancerous and so on, without killing them potentially. But also, while we are doing this, we might learn a lot of biology. Why? Because we will be failing. We will definitely fail. And by failing, we will learn from our mistakes. And this is what happened by trying to control non-living systems as well. We had failures, and all of those failures told us more about how things work. They told us about physics underlying control. So the same way, I think biology and medicine will advance in major ways if we learn how to control living cells. So let's see how we do it. And the best way to approach this is probably to learn from nature. How does nature do it? Because nature obviously has ways to control cells. Because our cells behave in a way that they form tissues, they make us human. How does that happen? Well, the basis is in the so-called central dogma of molecular biology. Each of our cells in our body carries a genome, which is all of the DNA we have, and that's packaged in these chromosomes I mentioned. They are pictured here. And these genes are segments on this long thread of DNA. What the cell does is takes the gene as a template and makes proteins out of these genes. So these triplets of letters, which are nucleotides, are used to create proteins. The cell has machinery to make copy and copy and copy of proteins. And these proteins have two features that determine how they influence cells. These two features are sequence and amount. So first of all, it matters what the sequence of the amino acids is. They determine how the protein folds, where it goes in the cell, what it binds to, what it does. But more than that, the amount of protein matters as well. So it's not just the sequence. How much you have matters as well. If you have more, of course, the protein can do more of its job. It's like a worker. If you have more workers, you accomplish more of your job. So what I'd like to say here is that the sequences and amounts of more than 100,000 proteins control living cells. And they control the uptake, processing, and release of molecules. They control cell division, motility, invasion, cell death. And of course, as cells do all of these things, they become blood cells, they become skin cells, they become brain cells. And that's how our body functions. By genes, templating protein synthesis, and proteins controlling all things that various cells do. And the totality of our cells make us humans. But all of this can go wrong. And the way it can go wrong is by errors in sequences or amounts. So for example, if cell division, motility, invasion, or cell death have errors because of proteins having wrong sequences or amounts, that can lead to cancer. Also, we are trying to treat cancer. If the uptake or processing or release of molecules changes, we can put in cancer drugs, but the cell pushes them out. And that's also done by proteins. There are protein pumps that push drugs out. And that's why cancer treatments fail. So it all relates to this. This is how biology controls itself. And by learning from here, maybe we are able to take some of that control over and achieve better medicine. However, as I said, I show this picture. This looks very simple. In fact, the reality is much more complicated. We have more than 20,000 genes in our genome, and we have hundreds of thousands of proteins that the cell makes from these genes. All of these are interconnected. They control each other in very complicated ways that are partly unknown. So this is why this task is not simple, just taking this and trying to control it. And this is where synthetic biology comes in. Synthetic biology is a new area. It's a new, simpler approach to biology. What does it do? Synthetic biology aims to design and build biological systems for predefined goals. For example, to control cells. As examples, genes have been assembled into new synthetic gene circuits that can function as switches, oscillators, logic gates, pattern generators, and so on. What these systems do is they accomplish functions relatively independently of the cell's own regulation, regulatory network. So that's why building things from components like this might accomplish functions that you could not do by simply reaching in and modifying native genes in the cell directly. And another interesting thing is that we can build these systems. We can build switches, oscillators, logic gates, and so on. But if we start to evolve them, that may give us insights about evolution of networks, of genetic networks, that would be very hard, again, with natural networks. And I'll try to show you examples of that. So let's see some examples of synthetic gene circuits. As I said, synthetic biology builds things for predefined goals. And these things are new. They never existed in nature. That's why this is synthetic, because humans built these based on specific purposes or goals. One of the first synthetic gene circuit that was built was this one. It's called the toggle switch. And it consists of two genes, which make two proteins. These are the genes here, LAC-I and TETR. They make two proteins. The cell makes two proteins based on these genes, and these genes can suppress the other gene. So TETR shuts down LAC-I production, and LAC-I shuts down TETR production. So they mutually inhibit each other. And more importantly, we can control the action of the proteins by adding chemicals to the medium in which the cells grow. So if we add this molecule ATC, that will prevent TETR from binding to the promoter of LAC-I, and thereby, it's like releasing the brakes, LAC-I will turn on. Likewise, if we add IPTG, it will prevent LAC-I from binding to TETR's promoter, so TETR will turn on. So as you see, we can basically switch this system by pushing it one way or the other, with one chemical or the other. It's almost like a wall switch. And to emphasize the similarity, it is in another way like a wall switch, because on the wall, if you push the switch down, it will stay down. You don't need to keep pushing. You just give it one push, it will stay down. If you push it up, it will stay up. You don't need to keep your hand on. The same way here, you add the pulse of this chemical, it will push the system one way, and you add the other chemical and push the system the other way, and it stays there. You don't need to keep the chemical on. And that's why it's called a switch. It's a toggle switch. And you can monitor this by adding fluorescent proteins, which are green and red. So you can see the cells actually changing color as you add these chemicals and flip the switch. Here is example 2. That's called a repressilator. These are 3 genes. I'm just showing the proteins here, which repress each other in a circle. So these are bacterial proteins again. They repress each other. If you put this together in bacterial cells, you will start seeing cells that oscillate. So for example, the arrow here shows a cell that turns bright and then dims and then turns bright and dims again. And it keeps oscillating up and down. This is a behavior conferred by the circuit. And importantly, this has never existed in nature. It never existed in the history of life before humans came around and said, okay, let's build a switch. The same way the toggle switch is a system that only humans built for the first time. These genes have never regulated each other in natural history. So this is synthetic biology. These are its capabilities. And these are bacterial cells in these examples. But of course, this field is already more than 20 years old, and many advances happened, including in various eukaryotic cells, such as yeast, mammalian, human cells. And here is where I'd like to talk about what my lab works on and how we are different from the general synthetic biology field. My lab builds and evolves protein-level tuner systems. Let's think of these as dimmers. I showed you switches. Imagine that we are building dimmers or fine tuners, just like you see on the wall. Why do we want to do this? Well, protein-level tuners, we think, should enable next-generation controllable gene therapies. As I said a few slides ago, the amounts of proteins matter, not just their sequences. If you have too much of a protein, you can get cancer. If you have too little, you can get cancer. What if we could control protein levels to be just at the right level to avoid that? Also, by learning to control things, we may be able to understand deeper how gene expression affects phenotype. And think of this as a control knob. So a dimmer is like a control knob where you can scan something. In physical chemical systems, it's normal to have control knobs which you turn and adjust the temperature, adjust, for example, pressure, or various properties. In biology, we don't really have that. And I'd like to say that the switches we built, not switches, the dimmers we built fulfill that role. So from switches, we actually went to dimmers. That's what our lab contributed. And one more thing we do is we actually evolve these synthetic systems we built. So that's another thing that's not typical to general synthetic biology. We propagate them over long times and watch their evolution. So we watch their Darwinian evolution and draw conclusions from there. So let's see some examples from my lab. We actually built a dimmer from a switch. The switch consisted of this TET-R repressor, which you saw in the first two circuits I showed you, the toggle switch and repressilator. This was there. It represses this other gene GFP. So by binding this to this DNA element called TET-O, TET-R shuts down GFP. It's like the brake that stops GFP production. You can control this by this extracellular molecule ATC that diffuses into cells. It's a chemical you can put into the growth medium and turn repression off. It's like lifting the foot off the brake. And when you want to do this, you might think, okay, this could be a good control system, but it's actually not. What you notice is if you try to control it at the middle level, which is shown here, the cells don't go to intermediate expression. Instead, they go high, they go low, they go all over the place. Their average may be intermediate, but the individual cells are not. So that's what we call noise or variability in cells. However, if you add one element, it's a negative feedback, basically adding this TET-O sequence in front of TET-R, so that TET-R shuts down itself in addition to the target gene, GFP, immediately everything changes. What you see is the cells become much more homogeneous, and moreover, their expression level, their greenness can be tuned now from low to high, just like with a dimmer switch. So that's how we built these systems way back, and we've been using them ever since in combination with other similar systems in various cell types. So this is the genetic dimmer switch, also called the linearizer. As I said, this is transferable. Originally, this was developed in yeast cells, as I showed you, but we can transfer these systems to human cells. This is the effort published in 2013, where we moved these gene circuits into MCF7 breast cancer cells. And you can see the dimmer feature here. The greenness, again, indicates protein level. As we turn on this ATC molecule here, you see the greenness in cells goes up, meaning it functions like a dimmer switch. Also, the tightness of these histograms indicates these cells are all very similar. They are following our control. So synthetic gene circuits can be transferred to other cell types, such as cancer cells, after careful optimization. This required quite some work, figuring out what promoters to use, how to change the actual genes involved in the system. But it works in human cells. And since then, we put them in many other human cell types and also other mammalian cells, and they keep working with some caveats here and there. Okay, so as I said, we use these systems to control gene expression, and I said that the cells become very homogeneous. They all follow our control, having similar protein levels. We wanted to use this property combined with another gene circuit. We built this other gene circuit, which has positive feedback instead of negative feedback. And as a consequence, it has high variability. So instead of cells all being similar, with this system, cells become very different. So I would call this a precise low noise system, the one I showed you. And we have another system, which I'm not going to go into, which is variable. Every time a cell is like throwing dice, having high or low levels of proteins in a sort of random fashion. And when we have this, we see cells like this. You see on the top are the noisy cells. They are the variable cells. On the bottom are the homogeneous cells with similar gene expression. Also, the histograms here, you see the narrow blue histograms corresponding to the precise control and the broad red histograms corresponding to the noisy variable control. We can measure the variability by this quantity called CV, coefficient of variation. And you see the red is over the blue for a lot of expression levels, meaning protein levels, which means that we can choose a protein level which is the same in average. So the protein level is the same in average. The average protein is the same in each cell population, carrying gene circuit 1 or gene circuit 2, both of which are integrated in different cell lines, but their variability will be different. So basically what we do is we equalize the means and we have different variabilities. Just like these histograms shown here, they actually have statistically indistinguishable means and different variances. Why is that good? Well, in the cancer field, there is this theme ongoing that variability aids drug resistance. But whenever you want to test this, there is a problem because you can make cancer cells more variable by various methods. However, the danger is that the mean will change as well. So how do we ensure that, let's say, drug resistance we see is attributed to variance? The best way to do it is to make means identical. And this is one of the ways, and one of the few ways you're able to do this. Having gene circuits which can have equal means, can confer equal protein means, but different variabilities. And then you can start asking, how does variability affect drug resistance? How does variability of cells affect drug resistance? So we set up cells just like I showed you with equal means and high and low variability. The blue cells have low variability. They're the low noise cells. The red cells have high variability. They are high noise cells. And then we added Puromycin. Importantly, this system, besides GFP, includes PuroR. PuroR is a Puromycin resistance gene. So it prevents the action of Puromycin, which otherwise kills the cells. It's a translational inhibitor. So having cells compete in various levels of Puromycin, we can ask who is the winner. This is like a survival game. Adding increasing levels of Puromycin, we compete the cells and ask, do the blue cells come back first, or do the red cells come back first, if ever? Well, first of all, for 0 Puromycin, so no drug, we see that the cells grow very similarly. We have 6 replicates of each, and they are practically indistinguishable. When we add low Puromycin, we see that the blue cells start growing first. They take longer. Notice it's longer before they grow up, and they are inhibited, but they come back first. The red cells take much longer, about twice as much as the blue cells to come back. So the blue cells are the winners of the competition at low stress. As the stress increases, it becomes more complicated. The blue and the red sets approach each other, and they become very stochastic. Individual replicates come back at different times. And this becomes even more extreme when we go to really high stress. So at high Puromycin level, you see that the blue cells never come back, and it's only the red cells that come back. So to conclude this, it turns out that variability, which is higher for the red cells, is advantageous for cells to come back at high stress, but not at low stress. At low stress, actually, homogeneity confers the cells advantage. So the role of variability as a message depends on the level of drug you use to treat these cells. At high drug levels, variability helps survival. At low drug levels, variability hinders survival. And this matters because cells in cancerous environments see different levels of drug. So this came out of this experiment, and it was an interesting conclusion. But we were asking how. How did the cells grow up? If you hit them with Puromycin without this gene circuit, they just die. So somehow the gene circuit allowed them to survive. How? So this is what we investigated. These are microscopy images of the cells early on. You see many cells as they see the puromycin, they suffer, many of them die. So we keep them for a long time. If you check the timescales here, it's about 2 months constantly in drug. But after 2 months, surprisingly, the cells come back. Even though you would think they are gone, some of them come back. And lo and behold, all of them have high GFP levels. And in this gene circuit, GFP, the green color, is coupled with puromycin resistance. So GFP and puromycin resistance proteins are both high. So the answer of how this happened is by overexpression of puromycin resistance. You add more and more drug. What the cells do is they create more and more defense. They create more and more of the puromycin resistance protein, which then cancels the effect of high puromycin. And that's interesting. One question we asked, is this stable? So after we remove the drug, do the cells keep their expression? And what we see is here, this is actually after treatment. And to illustrate what happens, how much expression increased, this is the expression level on this axis, compared to the ancestor, which is here with the dashed black line. You can see all of these replicates have very high expression. And then we did two things. We first of all removed drug, saying if this is transient, then the expression will drop down. There is no reason to keep high expression. There is no attack. There is no puromycin. Do the cells drop in expression with or without doxycycline? Because doxycycline was used here to induce the gene circuit. And the answer is, as you see, these blue data points are always way above the dashed line. I'm sorry, I'm having here. Okay, just a second. Let me reshare my screen. Yeah, the blue data points are always above the dashed black line for over a month. So this resistance of higher expression is stable. And then we asked how. So we sequenced the gene circuits, trying to figure out what the cells did to elevate expression of puromycin and GFP. And in 6 replicates, we found one where there was an explanation. In this one replicate, number 2, we found a TET-R mutation that broke TET-R, basically preventing it from acting as a brake, acting as a repressor. And if the brake is gone, of course, GFP goes up. So this explains why GFP was high and puromycin resistance was high in this replicate. However, in the other replicates, we had no explanation. We saw some unknown enhancer mutation in 2 of them, and we saw no mutations in 3 of them. So this was all unknown, and we did not know how expression could be high while TET-R being untouched, TET-R being perfectly functional. Okay, so we tried to look for native resistance mechanisms. Maybe the cell does something. Maybe these CHO cells, these are Chinese hamster ovary cells, did something in their native genome to resist treatment. So we did RNA sequencing, which would tell us at the transcriptome level, are there transcripts that go up and explain high puromycin resistance? And the results were surprising because these are all the transcripts we could map. And you see, this is the ancestor. This is the evolved sample with high puromycin resistance expression, and there's very little signal. So all of the native genes, they vary a little bit, but very few are significant. And even the ones that are significant, they are not even close to this cloud. And what we see in this cloud, it's all the gene circuit. All of the genes we put in, which were never in these cells before synthetic biology came around, all of these transcripts are highly expressed. And to see how much they are expressed is, you can look here. You see this gray level as the expression of the untreated sample. Compared to that, the evolved samples, the colored ones, are very highly expressed. So it's 50-fold or so on average, overexpressed compared to the untreated sample. So their expression is really high. And some of this makes sense. This is the plasmid we put in. It was genomically integrated. And you can see the genes, PuroR, this resists puromycin, EGFP gives the green signal, and here is TET-R. The same elements are here. Puromycin resistance is here. You see it's very high compared to the gray, explaining high puromycin resistance expression. But then you see GFP is high, of course, it's co-expressed. A little bit more surprisingly, TETR is high too, which is the repressor. So everything goes up in this series. Even though TETR is a repressor, it still goes up in expression. Even more surprisingly, there are other plasmid elements that go up without any obvious reason. There is this hygromycin resistance gene, which is for maintaining the plasmid in mammalian cells. It's highly expressed, even though we don't have hygromycin in these experiments. Also, some plasmid elements like near the origin of replication, or AMPAR, which is a bacterial selection marker, these transcripts are high. So we don't know what's going on, but looking across here, all of these transcripts being high suggests that this might be due to higher copy numbers of plasmid DNA. Even though originally we put in 1 copy and we verified that, because of growth or treatment by puromycin, the cells figured out to create many copies of the DNA we put in and thereby create higher expression. And the test for that is qPCR. So qPCR is a method asking, is DNA amplified? Do we have higher copies of all of these genes? And right here you see EVO2 is the one with TETR, NF is the untreated control sample. And you see these have that they have no amplification. They are the basal level of DNA. And in all the evolved samples that matter, DNA level is high. You see it's up to 25-fold higher than here. So in all of these genetic elements, including HyGR, which has no reason to be amplified, there is a high DNA copy number. So pure R expected, the rest less expected, but overall the whole circuit got amplified. And this amplification, which is the copy number shown here, correlates with GFP in this circle. So that means that the amplification is an explanation for the higher expression level of GFP and of puromycin resistance. So we saw this in data, but of course, we wanted to know where did these DNA elements amplify? Where are they? Can we see them? Are they chromosomal? Are they on chromosomes, or are they outside of chromosomes? So this is where KromaTiD helped us out. So thanks to KromaTiD, we started seeing where all these DNA amplifications happen. And we say seeing is believing, and that was really the case here. We really were amazed by seeing, with KromaTiD's help, DGH, we were able to see where these amplifications happened. And they happened in samples where we expected them. So the sample with TETR mutation, which didn't have DNA amplification, you see this one green spot, no amplification. In all the other samples, you see these multiple green dots indicating the circuit moved to various chromosomes, and they are brighter, meaning that this, there may have been tandem amplification. And you have all these stories going on in various cell lines like this, sometimes very impressive, huge tandem amplifications. And we also see extrachromosomal events in this replicate tree. So now it's a totally different story. We actually see what happened. We are convinced we have DNA amplification by various mechanisms on chromosome tandem repeats and extrachromosomal events. And that's where we asked, can we somehow use this to, these cells are drug resistant. So they are like drug resistant cancers, which where treatment fails. Can we do something about this? Considering that most of the replicates became resistant by DNA amplification. And you read the literature, and many of the cancer cells that resist therapy have DNA amplification. And in particular, this was not about drug resistance, but we found this study, even without drug treatment, some cancers have DNA amplification. And one typical example is HER2 positive cancers of breast and ovary. They have amplification of HER2, which is a membrane receptor. What this study did, we found in 2022, this study by Faye Rogers at Yale University and collaborators, showing that in HER2 positive cancers with high amplification, shown by these bars, this is the amplification numbers here. So BT474 4 has the highest amplification, really high. You see, they propose this therapy. That's by using DNA to couple up with amplified DNA, you can cause DNA breaks. And this is where I'm showing this happening. So the normal DNA is this duplex or double helix, as you know, since Watson and Crick. They determined the structure. It was a major step in learning how biology works. However, if you add another stretch of DNA, purine-rich sequences are needed for this. They will form a triplex. So it's no longer a double helix. It's going to form this triplex structure. And it may look interesting, but essentially it will break. If this TFO is long enough, it will create stress in DNA, so DNA will break. And of course, the more of these breaks you can cause, the more the cell will feel it as a damage, as a stress, and eventually it will die. So this is illustrated here. The reds are the TFOs in their paper. And if you have just a few, the cell will live. If you have many, coupling to DNA amplified regions, the cell will die. So taking that example from HER2-positive cancers with DNA amplification, we said, okay, why don't we generalize this to drug-resistant cancers with DNA amplification? And not just cancers, DNA amplification and DNA damage, response, and cell death are conserved across mammalian cells. So first we tried with our CHO cells, Chinese hamster ovary cells, where we had these gene circuits. So we designed these TFOs shown in black, targeting regions in our gene circuit, shown here. And we had 3. And when we added them individually, we saw signals. So we started seeing that TFOs tend to slow growth. However, when we added them together with Puromycin, which was the original drug, we started seeing stronger signals. So these drops here show that the growth rate of cells dropped significantly by adding TFOs in addition to Puromycin. So we were suppressing these resistant cells. Now we tried another thing. Why don't we add all 3? So we added just maybe TFO2. Let's add TFO1, 2, and 3 together. When we did that, the signals became really strong. So when we add 3, these TFOs that break DNA in 3 different places, in amplified DNA, where the signals get really strong, and they only affect cells with DNA amplification. That's what these dots indicate on the left. All right, so this was encouraging. Why don't we try it with cancer cells? These are Chinese hamster ovary cells with a synthetic gene circuit. It's a model. Let's see if it works on actual cancer cells. So that's what we tried. We took these H69 drug-sensitive parental cells, which were evolved to become doxorubicin resistant previously. And they became doxo-resistant by DNA amplification. Of this ABCC1 transporter, this is a DNA element coding for a multi-drug resistance transporter. And you see this is hugely amplified, this DNA, and that's why the cells resist doxorubicin. And you can see how doxorubicin affects the parental cells in gray much more than the resistant cells. So we added TFOs to these, and what we see is remarkable. Well, first of all, for the parental cells, we add these TFOs and, it doesn't matter. So they are suppressed by Puromycin. You see they go down. But the TFOs, the DNAs, don't matter. However, for the cells with DNA amplification, TFOs matter hugely. So they are resistant to doxorubicin. As you see here, doxorubicin indicated in the gray level doesn't affect these cells. But when you add TFOs, immediately you see a huge effect, which is already present even without doxorubicin. So overall, it seems like this DNA therapy works on cancer cells, and we with DNA amplification, and we verified this for another cancer cell line, which was 5FU resistant. It was colon cancer. So we think we have a lead here. We have an idea that this might be a general way to treat cancers drug resistant to DNA amplification. So what I'd like to say here, I think hopefully you saw how genome modification, synthetic gene circuits, give you access to precise protein level control, which is needed and useful. You can draw very interesting conclusions from that. One conclusion we saw is that heterogeneity does not always promote survival and drug resistance. And resistance due to DNA amplification can be targeted by a new form of nucleotide therapy. Also, I'd like to mention we have other areas of our research. We developed new post-CRISPR genome engineering method. It turns out that DNA structural rearrangements are important when you try to modify cells. And Aaron will talk to you about more on that. There's this recent method that avoids that DNA damage due to CRISPR-Cas that's been developed, and you're welcome to look at. So at this point, I'd like to mention all my lab members who contributed to this. This is a long-term effort, as you saw. It's been going on for decades, and many people contributed to the mammalian projects. These are lab members, some of them still in the lab on top, and some of them have moved on, but huge thanks to all of them. And of course, many thanks to all my collaborators, lab members, mentors, past and present, to funding. This was only possible because of NIH support, special support, because this is not very standard type of research. And many thanks to KromaTiD for making things visible. And it was a totally different experience. Also, the bioinformatics was done by Chien-Hua Mu from Hong Kong University of Science and Technology, who I met at the Cancer Systems Biology Conference, and I met Chris Tompkins CEO of KromaTiD at Oxford Global Conference in Boston. So I'm also grateful to that conference and their organizers. So many thanks for making time, and I'll give it over to Aaron at this time.
Thank you, Dr. Balázsi, and thank you to all the members of the audience for both attending and then bearing with us while we dealt with some technical issues at the front of this presentation. Very happy it got back online and we were able to hear Dr. Balázsi give his talk. We at KromaTiD are very happy to have contributed to this important work. I think your application and this project specifically really showcases some of the advantages of being able to look directly at what's going on in the cell with the custom tools KromaTiD offers. I thought I'd follow up first by walking through some of the images from your experimental samples a bit more deeply. For this project, KromaTiD designed what we call a custom insight assay to assess the specific target of interest, Dr. Balázsi's synthetic gene circuit in CHO cells. KromaTiD has a flexible suite of bioinformatic design tools which allow us to create background-free target-specific probes for use in any sequenced genome of interest, including the CHO genome. To measure Dr. Balázsi's target of interest, we designed and configured an assay made up of a genomic control probe for the CHO genome, seen in pink, alongside a probe designed to detect the synthetic gene circuit, which was about 8 kb in size, seen in green. This is a single cell from one of Dr. Balázsi's samples, showing the expected presentation of signals in a metaphase spread. The very small green signal shown enlarged on the right is the size of signal expected for a target of this size. I'd like to point out that the signals in these chromosomes are showing up on a single sister chromatid, which is a bit different than a standard metaphase fish signal, which will appear on both sister chromatids in a metaphase chromosome. For the DGH method, probes are designed directionally against a single parental strand in a metaphase chromosome, and when applied to cells that have been prepared for DGH with the daughter strands removed, the probe will only bind to one of the sister chromatids. For this assay, a genomic control probe was used as a confirmation that both the cell preparation and the hybridization was good, and to qualify the cell for analysis of the variably integrated synthetic gene circuit. Next. In this next sample, the green probe signal has a much different presentation. It's both substantially brighter, and it appears to be laddered on the chromosome and present in both sister chromatids, indicating the presence of large tandem and inverted tandem integrations of the synthetic gene circuit in this cell. This interchromosomal amplification event is readily visualized by the DGH Insight assay, and by analyzing many individual cells, the rates of these events can be specifically tabulated to map the amplification events and the heterogeneous presentation of the synthetic gene circuit in the samples of interest. Next. Another type of event detected by the InSight assay was the presence of the synthetic gene circuit on extrachromosomal DNA, or ecDNA. ecDNA is, in essence, a circularized chromatin fragment without a centromere, often containing amplified and decompacted genomic sequences. There's a strong correlation between ecDNA formation and genome instability, and several recent studies have shown that various molecular events associated with chromosomal damage can lead to the formation of ecDNA, including chromatipsis, religation of DNA, breakage fusion bridge cycles, and replication fork stalling, and also template swishing. Cancers that contain extrachromosomal DNA, ecDNA, are associated with aggressive tumor growth, drug resistance, and shorter patient survival. The high overexpression of oncogenes by ecDNA leads to the malignant growth of tumors. Mapping these events, their genesis, and their rate and their presentation in a dividing population of cells is of great importance. Here we can see the synthetic gene circuit of interest is present on an ecDNA fragment, alongside several other ecDNA fragments present in the metaphase spread. Rates of synthetic gene circuit insertion, intrachromosomal amplification, and association with chromatipsis and ecDNA were measured by the DGH InSight analysis performed in support of Dr. Balazsi's project. Next. Detecting and characterizing genomic integrations is only a small part of what KromaTiD can do to map structural variation, detect genomic instability, and measure heterogeneity in any dividing population of cells. Ultra-high-resolution data on single cells is useful and can be essential for identifying genomic structural variants and measuring heterogeneity and clonal outgrowth. On the left is another example of a custom DGH InSight assay, similar to the one used for Dr. Balazsi's project, but tracking the outcomes of 2 genomic CRISPR edits present in close proximity on the same chromosome, as opposed to an integration event. In this cell, an inversion rearrangement frequently seen in this system is visualized as a yellow signal, probe targeting the segment between the 2 edit sites, present on the opposite sister chromatid from the red and green signals, which target the 2 genomic regions flanking the edits. On the right, next, is our whole genome DGH screening assay, which can be utilized to measure structural variants across the cell in an unbiased way. Next. In this cell, you can see a small telomeric inversion on one homolog of chromosome 1 and an unbalanced translocation involving chromosome 19 and chromosome 17. Next. I'd like to wrap up by showing a couple more examples of use cases for KromaTiD assays, looking at both the DGH suite of assays as well as some of the FISH applications our assays fit into. Here, DGH screen is used to profile complex rearrangements in a cancer cell line. Aneuploidy, translocations, inversions, and other complexities, including presence of complex amplifications, marker chromosomes, and ecDNA, are all present in this cell. Cancer cell lines often have complex karyotypes, some of which are relatively stable and others that are not, and are rapidly changing and evolving as the cells divide. The screen assay can provide several types of data: variation from the reference human genome, variation within a sample, heterogeneity, and variation post-passaging or post-treatment, as compared to an earlier passage or pre-treatment sample. Next. Shown here is a KromaTiD pinpoint FISH probe designed to target an oncogene. There are 3 copies of this oncogene in this particular cell, which is karyotypically expected presentation in this cancer cell line. In this next slide, we see variable levels of ecDNA containing the oncogene of interest. One cell with relatively low copy number and another cell with a very high copy number, in addition to the chromosomal loci. Being able to detect and visualize these potentially deleterious amplifications and their incidence in treated and untreated populations is important and something KromaTiD's custom assays are uniquely suited to do. As demonstrated in this work with Dr. Balazsi. Next. Next, I'd like to shift gears and look at the genomic structural abnormalities associated with some CRISPR-Cas9 editing systems. These type of variants can arise in any gene editing system where double-strand breaks are generated alongside the background rate of spontaneous breaks present in every living dividing cell, and the repair process utilized by the cell is not homology mediated. For this experiment, T cells were transfected with CRISPR-Cas9 and guide RNAs for a single edit site located on chromosome 19. 2 probes, green and yellow, were designed to bracket the chromosome 19 edit site. In panel A on the upper left is a cell with the expected assay configuration, where yellow and green probes are co-localized on each homolog of chromosome 19. See enlarged just to the right of the metaphase image. Panels B through D contain examples of the genomic structural variants observed in this editing system. Panel B is an example of a target to off-target translocation. The signal on homolog B2 has split and is present on an unknown small chromosome enlarged in panel B3. Panel B1 is the other normal homolog. Panel C is interesting. It shows an example of one of the more deleterious events observed in this editing system, an unbalanced translocation between both homologs of chromosome 19, resulting in a dicentric chromosome containing both yellow signals, blown up in panel C1, plus 2 separate acentric fragments containing the green signal, enlarged and shown here below the dicentric. Unbalanced translocations, along with chromathripsis, are both known progenitors to the biogenesis of ecDNA. The fragment in proximity to the translocation breakpoints can be amplified, deleted, and circularized, resulting in the accumulation of ecDNAs. The 4th type of event shown here is an edit-associated large deletion and resection event. In panel D, one homolog of chromosome 19 has a deletion of a large region adjacent to the edit site, resulting in loss of the green signal on that homolog, shown enlarged in panel D2. Next. Here, we are monitoring edited sites in another CRISPR-Cas9 system, this time a dual edit, with single color probes targeting each of the edited loci. This cell shows the expected assay configuration with 2 homologs of each locus, no rearrangement. Next. To focus in on a few of the more complex outcomes that can arise from misrepair at edit sites, here's a cell with 2 reciprocal translocations. 4 red and green fusion signals are present in this spread. One of the reciprocal translocations has resulted in the fragmentation and chromathripsis of both the chromosome derivatives, shown enlarged and to the right. Next. In this second cell, a translocation event has resulted in a multiradial chromosome with evident gain. Cells with these outcomes may or may not survive and are frequently associated with elevated rates of both micronuclei and aneuploidy post-expansion. Next. And now I'd like to thank you for your attention, and to conclude, directional genomic hybridization falls into a class of metaphase probing or painting methods that may be used to map chromosome structure and structural variation. Shown here in cartoon space are KromaTiD's DGH tools. DGH Screen and DGH Discover are whole genome mapping methods employing millions of fluorescently labeled hybridization probes to map chromosomes or even entire genomes. DGH Insight uses the same DGH method, but to map localized structures of interest. For instance, rearrangements involving edit sites or the location of random lentiviral inserts throughout the genome. DGH Screen and Insight are available to researchers worldwide, and our first assays based on DGH Discover are currently in beta testing. With these 3 products, you can map literally any structures and structural changes to genes, chromosomes, or genomes using single cells in metaphase. For situations where this data is important, but cells aren't already fixed or won't divide, say hepatocytes, we employ pinpoint FISH, a technique that uses DGH Insight probes in an old school cytogenetic method. There are, of course, other old school techniques that can provide data on chromosome structures, but typically at a much lower resolution. G-banding, for instance, is a technique for staining the secondary structure and is a fantastic method for orthogonally confirming large structural changes, such as trisomy. But where we are today, resolving structural variants of 5 kilobases or less with DGH, techniques like G-banding have resolutions of 10 to 15 megabases. Applications where these tools shine and can provide valuable information for both researchers and drug development and release pipelines include locus-specific and whole genome genotoxicity assessment, heterogeneity assessment, clone stability assessment, as well as engineered cell drug product genomic assessment. There are also powerful tools for biomarker discovery and rare disease studies. I would encourage you all to head to our website, www.KromaTiD.com, for more information, and feel free to reach out directly to us if you have any questions about what we offer and how we can support your work. All right, I think we're going to switch to some Q&A at this point. And there have been a couple of questions that have come through. First question, and this is to Dr. Balázsi. How universal is TFO-mediated cancer cells growth suppression? Are triplex helix-mediated apoptosis proteins ubiquitous in all tissues and cell types?
Yes, it's an excellent question. So start from the last part. Are triplex helix-mediated apoptosis proteins ubiquitous? Yes. So these are not triplex helix-mediated apoptosis proteins. They are DNA damage response proteins. So whenever you have DNA damage, the cell faces a decision, repair or die, senesce and die. So it's the same, doesn't matter really. I mean, it matters a little bit. It's single cell break, single strand break or double strand break. But the type of break pretty much determines how the cell responds, irrespective of if it's TFO or UV or X-rays or stuff like that. So these are general responses conserved across mammalian cells. Of course, the first part of the question is excellent. Is TFO mediated cancer cell suppression universal? It's universal, I think, for cells that have DNA amplification. So you have to have proper high-level DNA amplification for this to work, because you have to create enough DNA damage. Relating to the other part of the question, if DNA damage is modest, the cell will repair it. It will try to repair it. You have to have enough DNA damage for the cell to undergo apoptosis. Actually, it's the same with radiation or other types of DNA damage. So the same applies here. That's why you have to have high amplification. And once you have that, you have to find the Purine reach stretches long enough to target by TFOs. These stretches have to be long in order to create the stress for DNA to break. And if you have many, that's when DNA breaks accumulate and apoptosis starts. Does that answer the question? This is under investigation, so we are on the lookout for other cancers and designing TFOs for other cancer types, trying to develop this actually further.
Thanks, Dr. Balázsi. There's a couple more if you have the time. Can you go into more detail about how you control the dimming levels? Is it possible to optimize a gene circuit system to get specific levels of a protein?
Great question again. The answer depends on where. So far we have done this in vitro in cell culture, in culture flasks. Yes, we can. Various cell types are controllable in this way. We inserted, well, we have actually a pipeline of developing cell lines for accepting gene circuits. This is what I mentioned in the last slide. We have a two-step method where we create a so-called landing pad in practically any human cell line in a safe harbor site, it's called. Once that's done, then we can insert and remove gene circuits as many times as we want. We can put in variable protein level controlling gene circuits or precise control circuits and so on. And yes, we are able to do this. Once that's done, then we use chemicals such as doxycycline to control protein levels. We have other systems for orthogonal control. We have a chemical called DAPG controlling a repressor called PHLF. It's also from bacteria. That's what's beautiful about synthetic biology, that you can use bacterial repressors to control human genes with appropriate inducers that have minimal side effects. Also, I'd just like to mention that you can use light to control these systems. We have these feedback systems controllable by light, and I'm happy to send references or talk about details here. But in animals now, in vivo, we are trying to actually collaborate on this, trying to do this in mice, in chicken embryos. It's something that we are very interested in, how good this control can be in a living animal. Also, we are trying this in organoids. So all of those answers are hopefully coming.
Great. I think there's just one more question. This researcher says, I'm particularly interested in the dimmer switch concept for regulation of metabolism. Would applications in genetic disease be practical, say autism?
Yeah, that's an excellent question. So yes, autism is, although the type of disease mentioned is a complicated one. So I'm not sure. Autism is so broad and so complex. It's not one of these simple genetic diseases. However, there are other ones that depend basically on one gene, basically on amino acid synthesis or things like that. And there are gene therapies being developed against that. Multiple diseases, I know of research ongoing at NIH. Also, another example is sickle cell anemia, which is the hemoglobin B gene. So gene editing is being developed to address that, which can be done in two ways. So first, you can correct a mutation. Second, you can put in a correct copy of hemoglobin B. Same way for metabolic diseases, you can put in a correct copy of the mutated gene. And the problem is when you put in a correct copy, is this expressed at the right level? And this is where I talked about proteins controlling cells in two ways. One is sequence and one is amount. And I think amount is regarded much less. Everybody's worried about sequence, putting in the right sequence, the correct sequence, but we worry less about how much protein is made from that template. And I think that's hugely important. So when we develop gene therapies, are we actually making the right amount of protein? Do we have side effects? Overexpressed proteins can have non-specific binding. They can cause aggregation. They can cause many, many things we don't even know about. And this is where I think it's important to scan protein levels and see what their effects are. So long story short, I'd like to say controlling protein levels will be important when we develop these novel gene therapies by inserting genes to maintain control and figure out what the proper level of expression is in a certain tissue or in a certain patient.
Thank you. I think that concludes the Q&A section, all of the questions that we've received. So I'd like to thank the audience for joining, and thank you, Professor Balázsi. You will all be receiving a link to the webinar in a day or so. We hope you will take a few minutes to fill out a short survey at the end. Thank you again.
Thank you so much. Thanks everyone for taking the time, and I hope you have a great day. Any questions, please get in touch. And many thanks to KromaTiD for organizing this and for going to the phrase "seeing is believing" helped us see it. Thank you.
Have a good day, everybody.
Have a nice day.